Hello class, good day. So I will be discussing to you the techniques in isolating pure culture in a way that we will be identifying correctly the species of the bacteria in the laboratory. And mind you guys, the techniques that I am going to tackle here are all manual techniques. So sometimes some of the methods here are already obsolete and they are no longer used in the laboratory. Of course, okay, we have the term here isolation. So that's very easy because that's just referring to a process whereby microorganisms are separated, okay, separated from each other and cultivated in a culture medium. So why do we do this isolation technique? Okay, remember if you are a nurse or maybe you have been working in the hospital or you had worked in the hospital before, normally, okay, the patient would just submit specimen to the laboratory, right? And that specimen, let's say sputum or urine sample, that specimen might contain thousands or even millions of microorganisms or bacterial microorganisms. So of course, okay, because of the number of the bacteria or other microorganisms present in that specimen, we need to separate most especially the pathogen to the normal flora which might be contained in the specimen. So that's why we need to isolate. Okay? And of course, our aim is to produce a pure culture. So meaning to say, we are only having a single species of microorganism. Okay? And maybe if you read a book, some of the terms that you can encounter also in isolation techniques or when you are studying about pure culture is the term stock culture. Because if you are doing research or maybe you are in the academe, normally they would stock a pure culture. They would keep it. They will store it, okay, in the recommended temperature, in the recommended environment, of course, to make sure that that stock culture, okay, only contains one single microorganism and is not contaminated. So stock culture. And then also we have other term, mixed culture. So how about this one? When you say mixed culture, of course, it's not a pure culture. Okay, meaning to say the Petri dish might contain several colonies. Instead of just seeing one color of colonies, you might see whitish colonies, creamy colonies, or maybe yellowish colonies. So by just merely looking at the colonies, you will understand and you will have an idea that what you are dealing with, okay, is not a pure culture. So that is a mixed culture. Okay, so how to come up with a pure culture? Of course, some of the techniques here were tackled the last time when you do the smearing technique, right? So number one is, of course, in order for you to come up or to make sure that you are isolating a pure culture, you always have to maintain aseptic technique. Okay, so examples of aseptic technique is number one, okay, flaming the wire loop. Okay, so let me just emphasize, I know that some of you might not be working on this again, but if ever that you will be dealing with medical technologists or you will be teaching other medtechs someday, at least, okay, the simple techniques that you have learned, okay, can also be adopted by those people. So, for example, you are flaming the wire loop. Okay, so you have here the handle of the wire loop, and of course, you have the loop here, right? So, in order to make sure that we are maintaining aseptic technique, we have to flame the entire area, this one. Okay, so from the wire, okay, this one to the loop, okay, and you have to make sure that the loop and the wire, okay, are colored red, so it's red hot, okay, to make sure that the pathogens there, if ever there is any, because you are using this loop multiple times, okay, are eliminated. Okay, another example of aseptic technique is making sure that before you open and after you close the Petri dish, you have to flame, okay, the mouth or the opening of the Petri dish. Okay, for example, so you have here the lid, okay, and also you have here the body of the Petri dish. Okay? When flaming the Petri dish, you have to make sure that the lid is on top. Okay? Because of course, there are pathogens present here that might be overlooked. Okay? So in order for you to make sure that you are really eliminating the pathogens there or other normal flora, you have to flame, okay, that Petri dish again. Okay? Lid up. Okay? Or lid on top. Okay? However, if you are refrigerating, for example, or putting the Petri dish in an incubator, so what do you have to do? So it should be, okay, inverse. So the lid must be at the bottom so that the moisture will go on the lid, not on the culture, because you have here the culture, right? Because this moisture, okay, can contribute, okay, to the presence of the microorganisms. So that is also one aseptic technique that you have also practiced last time. Another thing, when working on the benches, okay, it's necessary that you have to disinfect before and after you culture. Okay, just to eliminate all the normal flora, all the microorganisms present in the bench top. Okay, so those are just examples again of aseptic technique, and those are again necessary for us, okay, in order to obtain pure culture. Another thing, you have to use appropriate medium. If you are suspecting a gram-negative microorganism, then of course you have to use culture media that are supportive for the growth of gram-negative. If you are suspecting for a yeast cell, for example, or fungal elements, then you have to culture, okay, using techniques and using culture media, okay, that are intended for fungi. Okay, appropriate medium. And of course, you have to know the correct isolation techniques. And as what I have said, okay, to come up with a pure culture, we have some manual methods, okay, on how to isolate the microorganisms. So number one, this is very common and most of the laboratories are still doing this, okay, right now. So we have strict plate, we also have pour plate, and we have spread plate. Normally, the two of this, okay, are done in research or sometimes in food industries or maybe in some institutions where water and food are being analyzed. So pour plate and spread plate. Okay, so the first isolation technique again is the strict plate. So in this illustration, you can see that you have here the first strict, the second strict, and the third strict. Sometimes class, there are techniques which you can see 4 quadrants, so such as this one. Okay, so you have here, okay, another one in this case, this one, and lastly this one. So this one, okay, this is the first quadrant, second quadrant, third quadrant, and fourth quadrant. Okay, but because of course, okay, as what you can see, the space in the Petri dish is limited. So sometimes we only have 3 streaks. Okay, so this is what we call as multiple. Okay, multiple. Okay, multiple interrupted. Okay, interrupted streak. So that's the term there. And another thing, so how do we do this multiple interrupted streak? Okay, so of course, if you have the specimen, for example, urine specimen, so you get a wire loop. You submerge that wire loop in a urine specimen until the loop is filled with the specimen. Okay, the specimen is here. And then of course, you transfer the specimen in this area, first streak. Okay, and then using the same loop that you use to pick up the specimen, you do the streaking, okay, such as this one. Okay, and what do you do next? Because in this area, the first streak contains concentrated microorganisms. So what you're going to do is you have to change a loop. So you get another inoculating loop. And that inoculating loop will be used to spread the microorganism from the first streak. So you have to touch only, okay, this one, from the first streak to the second streak. Okay, as what you can see, it's only the first line or the first initial line that will pick up, okay, the microorganism from the first streak. Okay, because the moment you touch, okay, the other parts here from the first streak, then you are still, okay, accumulating the microorganisms from the first streak. So That's why you need to make sure that it is only, okay, this portion, okay, will be touched, and then the rest will not touch that area again. Okay? And what do you do next? Okay, do not change the loop when you are going to streak from the second to the third. All you have to do is to flip the loop. Okay? Flip the loop and then continue streaking. Okay? So that is again multiple interrupted streak. Okay? So if you do this one correctly, okay, because sometimes there are still areas, okay, on the third streak where the pathogens and other microorganisms are accumulated. So you will know that you have done this correctly, okay, if you have obtained this type of streaking pattern, or I mean if you have obtained single colonies at the last streaking area. So in this case, the colonies here are concentrated, so you cannot observe the elevation, you cannot observe the color, the surface, or any morphological characteristics that you need for the identification of the bacteria. Because again, in the first streak, the colonies are concentrated. Okay? So that's why again, when you touch, okay, you touch only this portion and then, okay, continue streaking. Okay? And then you flip the loop and then you streak. So that's why at the last area, you can already see isolated colonies. Okay, isolated colonies. So that is the multiple interrupted streak. Okay, you also have this one. Okay, this is in nutrient agar. So you can see here pure culture. Okay, one single colony. So if you want to identify what microorganism is this one, then you just have to touch one single colony. Okay. Another thing, you have here a multiple interrupted streak. However, this method is not that good because again, aside from the fact that this agar is corroded, okay, that agar is corroded, okay, this one doesn't have single colonies, okay, so you cannot pick up one single colony for you to do the biochemical testing, for you to do the identification. Okay, but what I am trying to project here is sometimes you might observe unique characteristics of the microorganisms. Okay, so in this case, this is a greenish metallic sheen from Escherichia coli. Okay, another thing, so this is showing the growth of colonies on blood agar plate. And if we will zoom this out, as what you can see, okay, you can observe hemolysis surrounding the colony. So instead of being red, for example, you can see clearing of the medium. So that is an indication that this microorganism is exhibiting beta hemolysis. Okay, and of course, this is still a good streak because you can see single isolated colonies. Okay, another thing, you have here the Serratia marcescens. Okay, so this is a typical growth of Serratia marcescens in the culture plate because the colonies, okay, are colored red because of the pigment prodigiosin. Okay, prodigiosin. I'm sorry for that one. Okay, prodigiosin, or others pronounce it as prodigiosin. Okay, so that's also one unique characteristics that you can observe. Okay, but it's not only Serratia marcescens that is exhibiting pigment production. You also have like Pseudomonas aeruginosa. Okay, instead of being color red, they can exhibit greenish. Okay, greenish coloration of the colonies and of the agar. Okay, because the Pseudomonas aeruginosa, okay, is producing pyocyanin and pyoverdine. Okay, this one, pyocyanin and pyoverdine. So that is again those are again the pigments produced by Pseudomonas aeruginosa. Another thing, so this is a parallel technique, okay, parallel technique recommended by Peltzar and Reid. So although this is a little more tedious as compared to the multiple interrupted streak, okay, the same principle applies. So meaning to say, you streak but it's not in zigzag, okay, motion. You do it parallel. You create parallel line. Okay, you have the first streak, and then of course you change the loop, and then you do parallel streak, and then flip the loop up to the last, okay, the last quadrant. So still, you will observe growth, individual growth, okay, at the last quadrant if you perform the streaking method correctly. Okay, another thing, this one. This is the lawn streak, okay? Lawn streak for urine specimen, but sometimes in layman's term, this is being called as Christmas tree, okay? Christmas tree streaking method because of how it looks like, okay? And this is usually recommended, okay? This is usually recommended for urine specimen. So it's easy because you just get one loopful of urine, and then what you're going to do is to make a straight line down, okay? And then you flip the loop and you start streaking in a zigzag manner. So in short, this is how the colonies would look like after incubation. So class, for bacterial microorganism, we usually incubate, okay, for 18 to 24 hours. So if there are microorganisms present in the specimen, of course, okay, you will see growth after this certain number of hours. Okay, but for fungal microorganisms, normally it would take 1 month for us to really see, okay, the growth of fungi. Okay, so this is again the lawn streak technique for urine specimen. So why do we do this one? Because in this technique, we are counting the colonies. Okay, we do colony count. Okay, and of course, it's not only that we do the counting, but we have to make sure that our inoculating loops are calibrated. Okay, calibrated inoculating loop. So if you are using 1 UL, okay, 1 UL inoculating loop, meaning to say the loop can hold 1 microliter of specimen. Okay, so the conversion factor is 1,000 for 1 UL. Okay, for 10 UL, okay, the conversion factor is 100. So 1 conversion factor 1,000, 10 conversion factor is 100. Okay, and this is the formula. So colony forming unit per ml is equal to the number of colonies counted times the conversion factor of the inoculating loop. So let's say, for example, you count the colonies here. Okay, so after counting the colonies, okay, you just have to multiply it with the conversion factor, okay, depending on the volume of loop that you have used. Okay, and how do we interpret? So if you have counted above or greater than 100,000 CFU per ml, meaning to say it is a significant clinical bacteriuria. Okay, so of course you have to report it. You have to indicate the count, and you have to put in the interpretation. Okay, however, if it is between 10,000 to 100,000 CFU per ml, okay, do not be confused, this one, it has a term above. So meaning to say 100,000 is still suggestive, okay, suggestive of infection. So ideally, okay, we would instruct the nurses or the patient to repeat collection of urine specimen using the first morning urine specimen because it's more concentrated. However, it still depends on the physician of the patient, okay, if he will let his or her patient repeat the collection or not. Okay, another thing, if it is just less than 10,000 CFU per ml, so it's not that significant, so it's contamination. So do not report it as contaminants or contamination. So just report it as negative or less than 10,000 CFU per ml. Okay, so that's for colony count. So for example, in this lawn streak, you have observed 50 colonies. Example. And then the loop is 0.001, okay, that's ml. So take note, 1,000 UL is equivalent to 1 ml. Okay, so that's why the 0.001, okay, ml, okay, that is again, okay, equivalent to a conversion factor of 1,000. So you use 1,000 conversion factor because 0.001 ml is equivalent to 1 UL. Okay, so if you counted, okay, 50 colonies and then you use 1 UL of loop, so you have to multiply the colony count again with the factor which is 1,000. So in that case, you have this interpretation. So 50 times 1,000, so that is 50,000 CFU per ml. So that is suggestive of infection. Okay, suggestive of infection. Another thing, okay, we have another isolation technique. So I hope we are already clear with the streaking technique, the multiple interrupted streak, okay, which is again commonly used in the laboratory to obtain pure culture. And of course, if we want to do biochemical testing for the identification of the bacteria, so of course, we pick only that single colony out of the streaking technique that we have performed. And also, we have the lawn streak technique for urine specimen for colony count. Okay, next streaking technique is the pore plate technique. So this one, again, as what I have mentioned, this is commonly used in research or in food and other industries, okay, together with a spread plate. Okay, so it is the pore plate. Okay, so this is an application commonly performed in the food, again, water, dairy, environmental, clinical, research, and pharmaceutical laboratory. So as what you can see there in the illustration, okay, you can still use this pore plate technique to determine the number of colonies per ml in a specimen, or the number of microbes per ml in a specimen. Okay. Another thing, okay, you might be asking, you just want to do the multiple interrupted streak. However, this one is a lot useful, okay, especially when you want to assay bacterial contamination on foodstuffs. Or another thing is that each colony here in this technique, okay, represents the CFU, okay, or the colony forming unit. Okay, so what do we expect here is unlike your streaking technique, the multiple interrupted streak, you cannot only observe surface growth, but you can also observe subsurface growth of colonies. Okay, and sometimes you can also observe that the colonies are embedded in the agar. Okay, so another thing you can also observe, okay, this is just based on experience. Okay, however, that one of the disadvantage, or one of the drawbacks, or one of the disadvantages of pore plate is that those embedded colonies, okay, you can observe that they are much smaller, okay, in size as compared to those which can be seen on the surface. Okay, so you have to really be keen enough, okay, to differentiate the colonies, okay, and of course in counting them. Okay, so this is how you do the pore plate technique. So normally, you would first perform dilution. Okay, so after diluting, okay, serially, okay, for example, in this case, you get bacterial culture here, 1 ml of bacterial culture, and you mix it with 9 ml saline. So 1 ml plus 9 ml. So again, the formula of dilution is the volume of what? The volume of solute over the total volume of solution. So that means volume of solute, which is 1 over, okay, volume of solute plus, okay, the volume of the solvent, in this case, the saline. So that means you have 1 over 10 dilution. So 1 is to 10. So meaning to say the first tube will have 1 is to 10 dilution. Okay, so since you obtained the same amount, okay, the same amount of specimen, or should I say mixture, from the first tube and transferred it to the next tube, so you have to multiply, okay, 10. So 10 times 10, that would give you 1 is to 100. And then times 10, that would give you 1 is to 1,000. And since you performed dilution, okay, you diluted the broth culture, you are expecting growth of microorganisms here, okay, very little as compared to the first tube and the second tube, or the first plate and the second plate. Okay, so that's the pore plate technique. Okay, so after you dilute, okay, you pipette the sample into the sterile plate, and then of course the culture medium will be added, okay, with the inoculum. But you have to make sure that the culture medium is not that hot, in order to not to kill the microorganisms, okay, if ever it is present in the sample. Okay, so that is again pour plate. Okay, how about this one? Okay, the spread plate technique. So the spread plate technique, so it's like also in the streaking technique, in the multiple interrupted streak, you are already using, okay, solidified agar. So you already have the Petri dish, okay, you already cook the agar. So instead of streaking using the inoculating loop, okay, you, what do you call this? You just spread the colonies, okay, on top of the, you spread the colonies on top of the agar. Okay, so that's the spread plate technique, and usually we use this L-rod, okay, or maybe this kind of spreader. Okay, so unlike your pour plate technique, okay, what you can observe, of course, are only, okay, surface growth. Okay, so this is the technique for spread plate. So we have the sample is pipetted onto the surface of the agar plate. Let's say you have 1 UL, or let's say 0.1 ml or less, okay, and then you just spread the sample evenly. And after spreading the sample using the L-rod, okay, you cover the Petri dish, and again you flame using a aseptic technique. And then of course during incubation, this is what I'm telling you, you have to make sure that the lid is, okay, below the body of the plate, okay, so that the moisture will not contaminate the agar. And of course you will see only surface colonies in the spread plate. Okay, so this is the comparison of the pour plate and the spread plate method. Okay, so in the pour plate technique, okay, you can dispense immediately the what do you call this, the diluted, okay, sample on an empty plate, while on the spread plate you have to pipette it directly on the solidified agar. Okay, and of course in the pour plate you have to add melted agar, and then you swirl slowly, okay, slowly, the Petri dish, okay, to prevent bubbles. That's why in the pour plate you can see surface growth, you can also see subsurface growth, and you can also see embedded colonies in the agar. Okay, for the spread plate, it's very easy. After putting the sample, you just spread using the L-rod, and then of course you can observe surface growth only. Okay, so this is the pour plate technique. Okay, another difference, okay, between your spread plate and pour plate technique, okay, is elaborated here. Okay, spread plate with agar, pour plate, sample first, and then melted agar. So the moment you pour the melted agar, of course, you have to allow it to solidify first before inversion or before incubating. Okay? So these are how the colonies would look like on a pour plate technique. So in this case, this is concentrated, okay, and then these are already diluted, okay, the one with excess. Okay? So technically, again, as what I have said, because you have diluted the last, okay, tube, okay, where you pour the sample, okay, on the agar, or maybe you mix it with the melted agar, is presumed to be having the least number of colonies. Okay, so you might be asking me, Miss, where can we, okay, start counting the colony? Of course, you have to find, okay, the least dilution or the lowest dilution where you can observe separated or isolated colonies. So in this case, you can count here, because of course you cannot count here. Okay, the colonies are still sticking to each other. So you can use this dilution factor. So you can count all the colonies on the plate, and then you multiply it with a dilution factor. So that's how you get the colony count, CFU per ml, or sometimes bacteria per ml. Okay, class? So those are just the isolation method. And let me just emphasize that the most common technique that we are using again is still multiple interrupted streak. And then we can also have the pour plate and the spread plate as optional isolation techniques in case it is really needed. Okay, it is really needed for the technique that you are doing, or maybe to the type of specimen that you are dealing with. Okay, so that ends my short discussion, and I hope that somehow you are enlightened, okay, with what I have discussed. Thank you.